This is an inhalation setup. For those of you who haven't seen one before, this was done at the Hamner Institutes in Research Triangle Park. This is a column where mice go into these little glass tubes, and they sit in there comfortably for about 6 hours. And then the nanotubes are nebulized over here and then go up through a tube, and then we can also tell how big the aggregates are and how much nanotubes they're getting exposed to and so on. There's a filter at the end of the collection port before it goes into the little inhalation duct where they breathe it nose only because they're obligate nose breathers, if you didn't know that. This is an electron micrograph of a filter, and it shows you that we're getting nanotubes on there, and we can collect them and count them. And there's a tube-like structure there and there. And we have then looked at where these go. So we've looked at the deposition patterns in the lungs of these mice after inhalation. This is another electron micrograph showing the deep lung, the respiratory areas of the lung. This is an epithelial cell deep in the lung. This is an air space. This is an air space. This is a nucleus of the epithelial cell, I believe. And this could be the nucleus of another epithelial cell, or it could be an endothelial cell next to a capillary. But if you look closely, what you will find are carbon nanotubes, small aggregates and individual nanotubes on the epithelial surface. So one of the interesting things we want to find out is where do they go in the lung after they're inhaled? Do they get into the deep lung? What happens to them afterwards, and do they do anything? If we blow up this little group here, we can actually see, quite fascinating, that these are intact carbon nanotubes that land on the epithelial surface here. Here's the air space again. This is the epithelium. And most of them land in aggregates, and some of them pierce into the epithelium like little swords. So what about fate and distribution of these inhaled nanotubes? They're cleaned up almost immediately by macrophages, these other cells. The macrophages are the amoebocytes of your lung. They're the free-moving cells that occupy everyone's lung by the millions, and they're your first wave of immune defense for inhaling pretty much anything into the lung. And nanotubes aren't unusual there. Any kind of carbon dust, any kind of soot you get from the street, macrophages avidly clean this stuff up and take it up the airways by something called the mucociliary escalator, this epithelium here, and you swallow them or they go through the lymphatics and you clear them. So here's a macrophage on an airway in the lung. This is the airway epithelium. This is blue staining collagen down here, and this is the airway lumen. And here's a macrophage that's chock full of carbon nanotubes. There's 3 more in the airway lumen, the opening of the airway. And in some cases, depending on what types of nanotubes you use, you can get some unusual effects. Tara was mentioning unusual effects in ecosystems. In the lung, you can also find some very odd effects. These are single-wall carbon nanotubes that form unique carbon bridge structures between 2 macrophages that tie them together. We don't really know what the significance of this is. We don't see it with multi-wall carbon nanotubes, perhaps because they're too large in diameter. We speculate that these single-wall carbon nanotubes are small enough, they're almost the same size as an actin cytoskeletal filament in a cell, which is about 1 to 3 nanometers wide and very long. And we think they may act like fishing wire and fishing line and get hung around these structures. That's all purely speculative. But these structures are never found in any sorts of exposures with carbon black nanoparticles. As a matter of fact, my research technician at the Hamner Institute's Jimmy Mangum, who originally discovered these, gave me a bunch of slides from nanotube exposed rats, a bunch of slides from carbon black nanoparticle exposed rats, and I could pick them all out blindly. I didn't get one wrong. These are a really reliable biomarker of exposure. It'll be really interesting to see occupationally one of these days whether someone, a human, goes in and gets their lung lavaged for a little bit of fluid, and we find these types of things. It could be a very important structural biomarker. So what do these look like inside macrophages at the nanoscale? This is a transmission electron micrograph that I find fascinating. This is a macrophage. Again, it's about 1,000 nanometers in diameter. This is the nucleus. This square blows up a collection of nanotubes. So after 24 hours and out to 2 weeks, these things maintain their structure. They don't get broken down very well, which is, I think, important. Macrophages are built to have acidic lysosomes where they package things very efficiently that they engulf. They wrap around things and bring them in. And they were believed that macrophages were evolved to take care of microbes that were encountered, and they still do today. And they're very efficient at breaking down things that are normally organic. Well, this is made of carbon, but they're not being broken down very well at all, as you can see here. They're still like long spears, they're hollow, and they maintain their structure very well. So if we focus in on the distal lung, where we're looking at the airways of the lung, and then these are the gas exchange regions. For this asthma model, we really wanted to look at the airways because asthma is a disease of the airways of the lung. So this illustration shows you an airway in cross-section so you can get an idea of what you're looking at in this histopathologic section if you're not a pathologist. This is a normal control lung. This is a lung exposed to carbon nanotubes. The blue stain is trichrome stain, which lights up collagen. So anytime you see blue, that's collagen, that's scar tissue. There's not very much here. As a matter of fact, this isn't really significantly different from normal. It looks like it might be. But on first glance, there's not a lot going on here with nanotube inhalation. This was my New Scientist quote. They didn't follow through and quote the rest of me. Here's allergen, ova. It's known to cause inflammation around airways, as you see in asthma. And then the combination of these two does cause some significant increases in airways. We can actually quantitate the amount of collagen around the airways by morphometry using digitized images. And here you see there's a significant increase in the amount of collagen around the airways with allergen and nanotubes versus allergen alone or nanotubes alone or aerosol saline alone. This will be coming out pretty soon in one of the ATS, American Thoracic Society journals. So what's the mechanism? I think it's important for us to begin to have some idea of if there's a toxic effect or we think there's a toxic effect. What are the mechanisms driving this? So what I'm told you is that nanotubes are taken up by macrophages. What we found, I won't show you too much of the data, I'll show you one panel, is that a growth factor, a protein produced by the macrophages called platelet-derived growth factor is increased. It is secreted. It binds to a specific receptor on another cell called a fibroblast. Fibroblasts are structural cells in the lung. They're the ones that deposit collagen. That's why they're so important in fibrosis. Platelet-derived growth factor, though, doesn't cause them to deposit collagen. It simply causes them to replicate, increase in number. Another factor, which is called transforming growth factor beta or TGF-beta, is increased by allergen challenge. And together, these two growth factors can orchestrate increased fibroblast number, increased collagen production, and that we're hypothesizing it contributes to the increased airway fibrosis we see with both agents, but not either one alone. There's the data that support this. Control, again, is saline alone. These are growth factor levels for gray as PDGF, open as TGF-beta. Nanotubes increase PDGF, not TGF-beta. Allergen increases TGF-beta, not PDGF, and both together could have a significant effect. So, inhaled nanomaterials, we're interested in how they cause disease, if they cause disease. Some are likely to be benign.